Okay. So I was asked to give a little bit of perspective on something that was brought up earlier today by Sarah Evans, and this idea of using a functional trait-based approach to understanding microbial diversity and complex microbial community assembly processes. And as Rob Knight alluded to, about 10 years ago, people really started to really improve the capacity to interrogate microbial communities, to build lists, catalogs of the biodiversity that are in a range of natural, engineered, and host-associated ecosystems, and even target functional genes to come up with essentially blueprints of how communities might operate. And I think the implicit assumption there was that we then take that information and like Janet was saying, come up with perhaps some more predictive models about how perhaps microbial communities might evolve, feedbacks between ecological and evolutionary processes, how communities assemble, the stability and functioning of these different ecosystems. But it's not entirely clear how that's going to happen just from a list of names of species. So a lot of people have been thinking in the general field of ecology and evolutionary biology that instead of focusing on names, we should be focusing on traits, functional traits. We could define those as physiological, morphological, behavioral, or even genomic properties of an organism or population that would confer some kind of, would have some kind of effect on the performance or fitness of those populations. And actually, so some of the work that first started kind of bringing this field from the general field of ecology and evolutionary biology into the world of microbial came from Jessica Green, who's here in the audience, back in about 2008. So people were already kind of thinking about moving away or trying to focus in on these functional trait properties. And I'm just going to kind of briefly go through some of the types of questions that we can ask if we start to think about and can identify certain traits of microbial communities. So one would be, can we detect certain predicted trade-offs that we know probably exist in complex ecological communities? So classic ones might be if you took a certain amount of energy that comes into a plant, that can only be allocated in certain ways. A certain portion of that's going to be allocated to above ground biomass, and a certain part of that's going to be allocated to roots. If we think about any kind of individual, there's going to be a similar type of energetic trade-off in terms of lifetime reproductive output and the longevity of that individual. So trying to identify some of these classical trade-offs would fall under the domain of a traits-based approach. Another thing we've heard about today is whether or not how we should be studying microbial communities. We hear some people who are characterizing communities based on the proportion of individuals at the phylum level, the coarsest taxonomic scale that we can think of. And oftentimes people will say, well, we should be looking at diversity at the O2U level, where we bin things at, say, a 97% sequence similarity. And we don't necessarily know which approach or which resolution is most appropriate for the question. Traits could be evolutionary, very conserved, or they may be very labile. And so this I think is this type of question is very important. I think it's ripe for thinking about in a traits-based framework. We can also think about whether a trait is, quote, a response trait or an effect trait. And so Scott brought up in the last talk the idea of biofilms. And in this case, a biofilm can actually function in both. It can actually confer a tolerance to certain stressors like metals, antibiotics, desiccation. But at the same time, when a microorganism allocates energy, exopolymeric substances, into a three-dimensional structure, it's modifying the physical environment in ways that are going to influence the species in its local environment. So this is what's referred to as an effect trait. And Jennifer Martinez is going to be talking more about this afternoon. So a lot of the approaches for, I guess the first thing I should say, there's a lot of challenges in adopting what's been used for plants and animals with microbes in some cases, even though it's very attractive to invent totally new theory. One thing, we can go out into a forest and measure the thickness or the texture of a leaf or the certain proportions or masses of individuals. And this is inherently much more challenging when we're thinking about microorganisms. We can't measure things always on an individual by individual basis. So we're often working with populations. If we want to know something about the physiology of microorganisms, often an easy approach is to start by working with isolated individuals or cultures. And of course, we know that we're still not in a position where we can do this with at least 99% of all the bacteria and fungi that are out there right now. So we're working with a restricted range of organisms to ask questions about mapping the niche or the physiological parameters that describe the population dynamics of a certain microbial organism. But I do think there are interesting new opportunities. In situ environmental enrichments can help us get at some of this, using signaling compounds to kind of coerce organisms into our culture collections, and perhaps using microfluidics to actually isolate individual cells. And so as some of you may have a background in microbiology, a lot of my colleagues will go into a population, they use something called reverse genetics where they'll knock out a gene and look for a change in a phenotype. I think there are also some opportunities where we can use those same genetic approaches and reintroduce those individuals into communities and ask questions about the community genetics of individual populations. So how do individual traits, for example, influence patterns of succession or assembly in different environments? And of course, we've heard a lot this morning, so I won't belabor the idea of using different types of multi-omics-based approaches or other types of sequencing technology. I would also say that there's a lot of opportunities that exist with using new visualization techniques. So Raman spectroscopy can get at isotope composition of cells, and so can nanosims technology. And so these are things that we should also be thinking about when we try to develop a traits-based framework for thinking about microbial communities. So I've just been thinking a little bit about some of the traits that maybe we could think about in a discussion at the break. What are the important traits that we should be thinking about for microorganisms in the built environment? So one I was thinking about is antibiotic resistance. Somebody also brought up the idea of toxins, so the ability of an organism to produce or not produce certain types of toxins. We also heard a lot about dispersal and colonization as being really important factors that influence the spatial distribution of populations and heterogeneous landscapes. You can imagine issues related to growth rate are obviously really important. And the last one I'm going to focus on for the rest of the few minutes that I have is this idea of persistence. So how long a population or an individual can persist in an environment under a given set of environmental conditions. And so we've been thinking a lot about this in the context of dormancy, which we define as a reversible state of reduced metabolic activity. So if a population or individual is challenged by conditions that are suboptimal for growth and reproduction, instead of dying, they can enter into this quiescent state. And there's really great examples of where individuals can lie dormant for, in some cases, millions or hundreds of million years in ancient materials like permafrost, halite crystals, amber. So the amount of time that organisms can persist is one that's very interesting. And again, this can be driven by environmental factors, but there can also be stochastic transitions that just some constant fraction of individuals will be allocated to this as a form of bet hedging. Of course, you need to be able to get out of this state for this to be an evolutionarily advantageous strategy. So that's something that we've been giving a lot of thought to. And there's a lot of ecological and evolutionary implications for this. Having a seed bank or this reservoir of taxonomic, genetic, phylogenetic diversity is really important for the persistence of populations, the outcomes of species interactions like competition, and the stability of ecosystem functioning. So just to kind of quickly go through a couple examples, one of the take-home messages is that in the environments that we've looked in, it can be variable, but in often cases there's a large proportion of the microbial cells in a range of ecosystems that are in one of these metabolic states of being inactive. So in soils, it's upwards of 90 to 95% of all the cells that are carrying out important geochemical processes, including trace gas flux to the atmosphere, must be only be carried out by 5% of the total number of individuals. That's still a lot of individuals, but there's this large proportion of individuals that at any given point in time aren't doing anything. We found the same thing, for example, in the skin microbiome. So somebody asked a question earlier in the last panel discussion about how we actually classify the metabolic activity of different organisms. Originally, we got into this by looking at 16S ribosomal gene sequencing and 16S ribosomal transcripts and found that those communities from a given sample look very dissimilar to one another. So since then, we've gone into trying to categorize the metabolic activity at a single cell level, either using epifluorescence or flow cytometry. In any case, if we look across these various different habitats, which probably vary in terms of the composition of the oils and fatty acid, moisture, salinity, exposure, dispersal, find that 90% of all the skin-associated bacteria can be classified as either dormant or dead. And so we've then used certain types of information and surveys to kind of build structural equation models to understand how lifestyle and demographic parameters influence those distributions. Really simple, basic microbiology that relies on using isolates and cultures. A couple of years ago, we decided we'd do a very basic experiment and just address the simple question of how long can bacteria persist? We know that in many cases, rapid reproducing bacteria can double, in some cases 7 to 15 minutes, things like E. coli and certain Vibrio strains. But how long can they persist in an environment? What's their longevity? And so we just took these populations of bacteria, spun them down and resuspended them in physiological saline. There was no inputs of carbon or any other nutrients. And we just tracked their population densities through time. And of the 28 populations that we saw, only one of them actually went extinct over about 1,000 days. So on ecological time scales, I think those are pretty meaningful duration of time to consider. With the remaining populations that persisted, there was about 4 orders of magnitude in variation in these net death rates, if you will. And we have some sequencing-based evidence to suggest that there are mutations that are accumulating. So these aren't just static cells that are sitting around the environment. There's still kind of some cryptic growth that's leading to the evolution of perhaps more resistant populations. And I just want to wrap up with one last example. When we started thinking about this life history strategy of dormancy, in some cases we know from model organisms what genes underlie the transitions into and out of dormancy. And so one classic example comes from Mycobacterium tuberculosis, the causative agent of TB, which can be dormant inside somewhere near 1/3 of the world's population. And there's a gene called the resuscitation promoting factor, which is very similar to, well, it lyses the beta-1,4 linkage of peptidoglycan, the major cell wall component of many bacterial cells. And so it hydrolyzes this linkage and it causes the bacteria to wake up from dormancy. And so we started screening some of our soil isolates and found that many actinobacteria, which is a dominant member of the soil bacterial community and also the skin, have this gene, and then we could express this in E. coli and basically had an anti-sleeping potion which we can actually go into seed banks now, ask questions about resuscitation, how seed banks might affect assembly processes and the function of those microbial communities, and also interactions with other macroscopic organisms. So with that, I'll just wrap up there, and I think we're going to have questions with the panel. So thank you.
So next up is Peter Dorston, who's at UC San Diego and a real pioneer in mass spectrometry and measuring metabolites, and we'll be continuing this discussion on functional interrogation, I believe.